Assalamualaikum. Today is our next lecture where I am discussing about gametogenesis and different type of eggs in the different vertebrates. Here, first of all, we discuss about gametes. Gametes are the sex cells. They are haploid in nature, in which they have the half number of chromosomes, egg and sperm. Egg is also called ova. Single is called ovum, and egg is large in size, and egg is non motile, which means non-moving, in which the yolk is present or absent. The yolk is present in case of amphibian, reptile, birds, and egg-laying mammal. It is absent in case of placental mammal. This one is the structure of a typical sperm. This is the head. The tip of the head is called acrosome, lysosomal in nature. This is the nucleus, and this one is called middle piece. Contains mitochondria that provide energy for the movement, and this is the tail which is used for the movement. And here is the structure of an egg. This is the structure of mammalian egg, also called oocyte. Egg is also called oocyte. Egg is haploid. This one is called nucleolus, and this is the nucleus. This is the cytoplasm, which in case of placental mammals, the yolk is absent in this area. Outer membrane is called zona pellucida, and outside the zona pellucida, these purple color cells are called follicle cells. They provide the nourishment and protection to the developing egg. Here is the first polar body, and these are the some points. This is gametogenesis, means the formation of gametes. Process is meiosis. Meiosis is a reduction division in which the chromosome number is reduced to one half of the parent cell. The product of meiosis is 4. The product of mitosis is 2. Meiosis occurs in the gonad and germinal layer. The germinal layer are the cells that produce the sex cells, that is the egg and sperm. Sex cells are egg and sperm. Sex organs are gonads, that is testes and ovaries. Somatogenesis is the meiosis that occur in the testes. Oogenesis is the meiosis that occur in ovaries. Meiosis occur in two steps, meiosis 1 and meiosis 2. Meiosis 1 is a reduction division in which the crossing over takes place and the pairing of homologous chromosomes occur. In the meiosis 2, there is no crossing over, there is no recombination, and it's just like a mitosis. As a result of oogenesis, in case of females, only one functional egg is produced and three are polar bodies which are discarded. And in case of male, all the four products of the meiosis are functional sperms. This is the process of meiosis. This is the spermatogonia, that is the sperm mother cell. It undergo mitosis to produce a large amount of spermatocytes. This step is called proliferation, means the basic function is to produce large quantity of primary oocytes so they can undergo the meiosis. Meiosis 1 results in the formation of two cells called the secondary spermatocytes. Then they undergo meiosis 2 and the four spermatid cells form. All of these undergo the maturation step called capacitation so that they can obtain their particular shape, particular structure, that is the head, middle piece and the tail. This helps them to penetrate the egg and fertilize the egg. This is called oogenesis. This is the oogonium, that is the egg mother cell. It is diploid, undergo mitosis to produce large number of primary oocytes that will undergo meiosis. Meiosis 1, as a result of meiosis 1, first polar body release. As a result of meiosis 2, second polar body release. And these meiosis, they take place after puberty, after maturation, and this first step takes place before the birth. Fertilization is the fusion of gametes. This one is the sperm, this one is the egg. They will fuse and they form a zygote in which the number of chromosomes is diploid, half from the sperm and half from the egg. It can be external, it can be internal. So first step is that encounter of the gametes. That means they come close to each other. Second step, they fuse, and their fusion is just like a lock and key method, and it is species specific. It means that a particular sperm of a particular species can fuse the egg of that species. Capacitation, the process in which the sperm become able to fertilize an egg. It gain all the properties to fertilize an egg. And egg releases a chemical which is called fertilizing. This activates the anti-fertilizing which is released by the sperm of the same species. This fertilizing and anti-fertilizing reactions help to recognize the sperm and egg. Next one is the sperm penetration. For that, the sperm you produce a lysine, which is called sperm lysine. It is an enzyme produced by ectosome. Ectosome is lysosomal in nature, present in the head of the sperm, and this helps the sperm to dissolve the egg core so that it can enter into the egg when the egg and the sperm nucleus they fuse. After their fusion, in other words, after their fusion, the formation of zygote occur, and when the zygote form, the egg become activated. Egg activation leads to the formation of fertilizing membrane. This membrane prevents the this membrane basically form around the egg, and it prevents the egg to fertilize to more than one sperm. When one sperm is entered, then this membrane prevents the other sperm to enter the egg. And another changes is ionic changes. The charge changing, the polarity within the egg become changed so that it prevents the further sperm to enter. And the next one is called preparation for the cleavages. Egg prepares itself for the cleavages. For that, egg produce its materials including mitotic apparatus, spindle fibers, and also the DNA. Fertilization is of two types. First one is external, that takes place outside the body of the female in the environment, that is usually water. Fishes and amphibians do this process. They release their gametes into the water where they fuse. But this method is not helpful as they have to produce lot of gametes. And the next one is that mostly their gametes are wasted by the water currents, by the environmental conditions, by the salinity, by temperature, pH, and also sometimes, but and sometimes they are eaten by the animals. So this is the advantages of external fertilization. For that, they do not need parental care, and they do not need to form protective layer around the egg, and they do not require any organ for the insertion of gametes to the female body, and the chances of fertilization they are enhanced by their courtship behavior. Courtship behavior means the behavior before breeding, in which the male and female attract each other by different physiological behaviors, and also that because of the external fertilization there is no need of much energy, so mostly the organism do this. And disadvantages is that gametes are usually waste because of the water plant. In time of fertilization, male should have a particular organ which is called intermittent organ to transfer the gametes into the body of the female, and usually in or near the reproductive tract where the fertilization occurs. This is again I said that will require the intermittent organ. These are the copulatory organ. What is a copulatory organ? The organ which is present in the male that helps the male to transfer the gametes to the body of the female. And this is the egg type. I show you a generalized egg structure. I want to show you that each egg has two poles, that is animal pole and vegetable pole. Animal pole containing nucleus of the egg, vegetable pole contains the yolk. And this is the egg of birds and reptiles. This is actual egg, which is basically the yolk. Outer membrane is called white line membrane. This one is called the cell, embryonic cell, that is the blastodome. Blastodome are the cells that are formed as a result of cleavages, and blastodome is basically the layer of blastomere is called the blastodome, and this is the airspace where the baby developing embryo can respire. And this one is called chelaza that helps the yolk to keep in place. And this is the albumin, that is the egg white. It provides the watery environment to prevent the desiccation or dehydration. Outer membrane is the shell membrane. The shell is basically made up of calcium carbonate and shell contain lot of pores so that air can easily enter into and out of the egg. Eggs are basically two types according to either they have a shell or not. The shell containing eggs are called cladoid and they are called non-cladoid eggs. Cladoid eggs are the eggs which have the shell, and shell is present in case of reptile, birds and egg-laying mammals, but it is absent in case of amniotic, in case of fishes, amphibians, and also in the placental mammals. This is the category of the egg according to the extraembryonic membranes. If the extraembryonic membranes, that are the membranes present around the baby, if they are present, that is amnion, which is a water bag. Chorion is protective layer. Allantois is used for the waste product storage. These membranes are present in the organisms are called amnions. There are the three types of amnions. Reptile, birds, and mammals, they are amnions. And non-amniotic eggs, these are the eggs that do not have extra embryonic membrane. There is no amnion, there is no chorion, there is no allantois. That is the fishes and amphibians. In such an egg development take place in the water. This is amniotic egg. Outermost is the shell, then chorion. This is the yolk sac. It contains capillaries that provide the food to the disc developing baby, developing embryo, and there is a water bag around the embryo that is called amnion. It provides the uh, moisture and also prevent the desiccation, and also provide the protection of the baby to the mechanical injury. And here is the allantois that store the nitrogenous waste, just like a baby excretory system. And this point where the chorion and allantois fuse to each other, this is called chorioallantois. This junction point is used for the respiration. And agar of two types, that is determinants and indeterminants, according to their development. The determinants eggs are the eggs which know their fate, in which the different parts of the egg know they say that which part of the egg develop into which organ into the embryo before their cleavage, before their fertilization. And indeterminate does not know that. Determinants X, they are present in case of protostorms, all the phyla except Echinoderm, hemichordate, and chordates. While in indeterminate X, these are the eggs that are present in case of hemichordates, echinoderms, and the chordates. So indeterminate eggs, if such a eggs they are divided after the first cleavage into two cells, into two plastomere, then the two complete embryo will be formed, and this due to the formation of identical twins. Good luck.